I can trust ConferZoom to upload the video automatically. I haven't seen it happen, so I don't trust it yet. So I'm gonna record locally. I'm going to move to sharing my screen with y'all. That can go away. Okay. No. Can you guys see anything of what I'm doing right now, out of curiosity?
Yeah.
Yeah. How I'm messing with Zoom right now, or just my?
No, just we see chapter 4.
Okay. 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 Cool. All right. So I've got 3 windows open here. I've got my PowerPoint, and I'm going to be like swooping back and forth between PowerPoint, between Safari, which has Canvas. I just like Safari, whatever. I have Safari open, and then I also have Firefox open for Wiley Plus. Since Wiley Plus only plays well with Chrome or Firefox, I just have it open in a separate browser. I hope that doesn't freak you guys out. So that's 3 different things that I'm going to be basically shifting back and forth through. So let's start with epithelial tissues, or about all the different types of tissues. So obviously we've got 4 different types. You guys are probably like, what is this already? Go ahead and mute, everybody. I've got weird noises now happening. Can you guys still hear me?
Really behind that other noise.
Yeah, but you guys can hear me, right?
Yeah, I can hear you.
Good unmuted that could still mute their microphone. See if we can get that to stop. If anybody hasn't clicked the little microphone in the lower left-hand corner, go ahead and click that.
Oh no, it's a lot of people.
Oh, I think we might have got it. Okay. Okay, cool. All right. Sorry for the interruption. Okay. 4 different types of tissues in the human body, okay? So these 4 tissue types account for all of the various tissues in our body. All of the cells in your body are part of one of these types of tissues, okay? Epithelial tissue is what we're going to go through first. It's going to cover all of the different boundaries between different environments. So it's a lining tissue. So it includes like your skin. It also includes the lining of your gut, because remember, we're a donut. So the outside of your body and your digestive tract are both like still the outside of the donut, right? The skin of the donut. So your epithelial tissue is both of those linings, making the boundary between the exterior world and the internal part of your body, the dough of the donut, if you will, and also other hollow organs. The functions of epithelial tissue include obviously protecting us from external environment, as well as secreting things. So your skin, of course, secretes oil. Your digestive tract is going to secrete digestive enzymes and bile and stuff like that. It also is going to absorb. Your skin absorbs moisture. Your digestive tract absorbs nutrients and water. And then, of course, filtering as well. And epithelial tissue is also found in some really important it lines certain tubes, so those other hollow organs that we just talked about right here includes parts of like your kidney and different glands that either like your kidney, for instance, filters things in and out of your blood. And then glands are going to secrete things, of course, into your blood or out onto an external body surface. The connective tissues of our body include pretty much everything that is in between the other types of tissues. So you've got epithelial tissue making the lining, your muscular and nervous tissue are there, and then your connective tissue is like in between all of those. So your connective tissue is involved with supporting all of our organs and all of the other tissues and keeping them in place. It protects them by making a squishy barrier. Connective tissue includes adipose tissue, so like our fat layer is protective, right? And it binds the other tissues together. It holds other tissues together. So in between the epithelial tissue of your skin and the muscular tissue of your bicep is connective tissue, right? So it's padding. It also includes cartilage and bone, okay? And other like tendons is also connective tissue. So we're going to talk about all those different types of connective tissue, the squishy ones, the cartilaginous ones, like stuff in your ear and stuff, and bone tissue, which is actually super cool when we get there. Muscular tissue, there are 3 different types of muscular tissue that we got to know. And they're pretty easy to differentiate once we get there. You've got skeletal muscle, which has its own set of identifying characteristics. Cardiac muscle, sorry, I could probably be like, here. I saw this, I was so excited. My laser pointer, it's back. So you've got skeletal muscle, you've got cardiac muscle, which is a different type of muscle tissue that is specific to the heart. And you've got smooth muscle tissue, which is the 3rd type of muscle tissue that is found in the walls of your hollow organs. So your digestive system is constantly pushing food through your body, right? And it does that with the help of smooth muscle. So there's a specific type of muscle that does that. So skeletal muscle, cardiac muscle, and smooth muscle are the 3 types of muscular tissue that we'll talk about. There are several types of epithelial and several types of connective tissue. These are going to be the big pains in our butts collectively, and we will go through them in detail. So don't worry. The last tissue type is nervous tissue. So this is, of course, going to be your central and peripheral nervous system. So your brain, your spinal cord, and all of your sensory and motor nerves, which is basically neurons and supporting structures. So that one is, it's pretty simple when we get there. So epithelial tissue, there's like 6 different types, plus a few more. Connective tissue, or sorry, I think there's just 6 different, no, there's a couple extra ones. So about half a dozen epithelials, about half a dozen connective tissues, 3 types of muscle tissue, and 1 really type of muscle or nervous tissue that we're going to talk about. Okay, are there any questions so far? We're doing okay? I'm assuming that you can all still hear me. That's what kind of freaks me out. Like I may have lost contact with you guys like 5 minutes ago and I wouldn't even know. All right, no, I'm assuming that you're still here. Oh, I can hear you. Yay, thank you. I love getting a little signs of life maybe when I ask if you have any questions, like unmute and say no if you don't.
I was wondering about the epithelial tissues because I know that I was trying to study them all and there's just so many.
There's so many, I know, it's crazy. We're gonna break it down, starting with 4 different types, and then we're gonna break down epithelial tissues.
Okay, perfect.
Okay, ready? Let's do it. Okay, so epithelial tissues. Let me real quick. Sorry, I want to make some adjustments here to my own window.
Like TTS order.
Okay, you can move your Zoom stuff to the bottom of the page too. There's like a little pointing down arrow next to the ID. I don't know if you guys have that since you're not the host, but I'm just rearranging my screen here so I can see everything that I want to see. Okay, epithelial tissue. We describe or we characterize and we identify different epithelial tissues by their function and by their location. So and by their structure, to be fair. So in terms of location, we've got epithelium so epithelium is singular, right? And epithelia is plural. The epithelium that is lining and covering body organs and our body as a whole and protecting it from the external environment is one main type. And then you've also got glandular epithelia, which is epithelial tissue that is specifically found in glands. And for that reason, it has important functions in secretion. Okay, let's talk about, I'm hoping this, oh, come on. Oh, sorry, I don't know what happened there. Okay, characteristics of epithelial tissue. So you've got two different locations where you can find it as a lining covering external parts of your body or parts of your body that are exposed to the outside world, and glandular epithelial tissue. All epithelial tissue has an upper and a basal surface, okay? All of the cells, they have this polarity. So polarity as in they have two different sides. So the top side of all epithelial tissues is called the apical side. It is the upper or free side. At the bottom, underneath the cells, of epithelial tissue, is the basal surface, okay? So it is the base that the cells are attached to, okay? So epithelial tissue, if you see that there's a part that is open to the air, and then a part, so like on a line of cells, one side of the cells is open to the air or whatever fluid, and the other side of the cell is attached to a base or like a base of connective tissue usually, but a base of some other type of tissue. That is going to be an epithelial cell. The apical surfaces of epithelial cells may have microvilli, which we haven't talked about yet, but that's basically little, actually, let's be smart and I can actually take you through the textbook here, which would be pretty sweet. Let's see if we can get this going. Oh, hello. Oh, this is so nice. It's gonna stop working in a second, I bet, just because I said that. Okay. So here's our 4 different types of tissues. We're talking about epithelial tissue. You can see just in this picture that you've got an apical surface and a basal surface of this tissue type, which is unique to this type of tissue, epithelial tissue. You don't see that with connective, muscular, or nervous tissue, okay? Microvilli, here we go. This is a picture of some cell junctions here, which we've already talked about. But it also includes some microvilli here, which is basically just like little outcroppings of the cytoplasm that just basically increases the surface area of the apical surface of that of the cell of that epithelial tissue. So that's what microvilli are. And that's as opposed to cilia, which epithelial tissue can also have, okay? The microvilli, which increases the surface area of that apical surface of the cell, is those types of cells. That type of epithelial tissue is found in the intestinal lining. So that increased surface area that the microvilli gives those cells basically allows them to extract nutrients faster than it would if it was just a flat surface, right? Increase that surface area, you've got more surface area in contact with your food, you're digesting food, and you can pull nutrients out faster that way with all the surface area. And then other epithelial cells, similarly, this apical surface of the cell can also have cilia. So this example, it looks like it could be microvilli, but it can also be cilia, which we have talked about. It is basically like hair-like projections that allow the cell to either move itself, which is not the case when we're talking about epithelial tissue. In the case of epithelial tissue, cilia is used to sweep things past the apical surface of the tissue, okay, of those cells. So if these were like little cilia, then they would all be sort of like flowing and sweeping substances past that apical surface of the tissue. So this is often found, or it is found in your trachea. So whenever you get a phlegmy cough, right? If you get a dry cough, then you need to pay attention. But if you have a phlegmy cough, or if you inhale dust or allergens, those little cilia in the cells lining your trachea are going to sweep mucus and the debris or whatever up and out of your lungs, right? So that you can more easily cough it out. The only other place that it is found is, maybe not the only other place, but one of the other major places that it's found is in the lining of the, God, I can't think of it. The uterine tubes, your fallopian tubes. So they sweep the egg that just came from the ovary down your fallopian tubes into the uterus, right? So they are polar. The apical surface can have microvilli or cilia, and the base, the underneath surface of epithelial tissue, so this is the apical surface and this is the base, the basal surface underneath, is often connective tissue, for one thing. It also tends to include glycoproteins and collagen. So remember, connective tissue is holding stuff together, so it's holding this epithelial tissue to whatever the underlying tissue is down here. Okay. Epithelial tissue is composed of closely packed cells. So the reason why your textbook immediately goes into cell junctions, which we had already talked about when we talked about cells, is because these cell junctions are specific to epithelial types of tissue. So epithelial tissue is tissue that has cells that are all very closely packed together and have cell junctions of various types, right, which we had already talked about, that hold them very close together. These other tissue types, connective, muscular, nervous. Muscular, maybe they have a little bit of cell junctions, but for the most part, they don't have these tight junctions where all the cells are really close together. Instead, you have, in the case of connective tissue, random cells floating around in an extracellular matrix, right? In epithelial tissue, they're all very, very closely packed together. And they all have cell junctions of one type or another, depending on the function, the purpose of that epithelial tissue. We already talked about the different types of junctions. Tight junctions make it watertight, right? So it keeps things from sneaking through. We want that in our digestive lining, so that you don't have like your food stuff getting through in between the cells like that. That would be bad, right? And then you've got like weird stuff floating around here. You get inflammation. That actually may be a cause of like irritable bowel syndrome, or like leaky gut syndrome you may have heard of. Is because of these tight junctions aren't doing their job, right? You can also have desmosomes, which we talked about. We described those as being like little tack welds. So they're really important for types of epithelium that is under mechanical stress to keep those cells together. So like your skin is going to have desmosomes between those cells of the epithelial tissue of your skin. And then of course you've got gap junctions, which allow things to pass from cell to cell, which is important for most, if not all, different types of epithelial tissue. Okay. Your epithelial tissues are supported by connective tissue, right? That is what the basal lamina is. And epithelial tissue is avascular, so there is no blood supply to epithelial tissue. That is a rule. Epithelial tissue is avascular, but it does have nerves. It is innervated, okay? So no blood vessels, nerves, yes, blood vessels, no. And it tends to have a very high rate of regeneration. So because it's exposed to the outside world, your skin and the lining of your gut, it regenerates really, really fast. So because it's under the stress of being exposed to the outer environment, those cells renew constantly, and that's why you're like constantly sloughing off skin cells, right? Because those cells tend to regenerate very, very quickly. Okay. Are we okay so far?
Yeah.
Yeah. Okay. If you guys, I don't know, if somebody's got like got a TV on or something, but
Yeah.
you can mute in between. That'll help us all with being distracted. I don't want to
I
name
have a
names or anybody,
question.
but yeah, question, go for it.
I'm trying. Okay. On the last, can you go back once of that?
One slide.
Yeah.
Sure.
This is supported by the connective tissue reticular lamina. So what is exactly, is
We're
it just,
going to talk about reticular lamina. We're going to talk about reticular connective tissue in a little bit. So that's just a specific type of connective tissue. I'm not worried about you guys knowing what this term means. The reticular lamina, particular. It's the basal structure underneath epithelial tissue, and it's made up largely of reticular connective tissue, which is just a type of connective tissue in particular. Yeah.
Okay. Yes. Thank you.
No problem.
I also I had a question too. So you said that for tight junctions, they're in, if I'm correct, you said that they're with the how do you say? I'm sorry, epithelial.
Epithelial.
Sorry, guys.
No, it's good. It's good that we have to say it out loud too, because that'll help you to like spell it and recognize it as well.
But within those tissues, you said that they mostly have, that's where the tight junctions are specifically for that.
Epithelial tissue is where you're going to find all of these different junction types. Yeah. The junction types are not as important in like connective tissue. I think there are some junction types in muscular tissue, but we don't really talk about them. We're not going to talk about them. Concentrate on epithelial tissue if we're talking about they're the types of tight junctions or desmosomes or gap junctions that we've talked about in terms of holding cells together. That's really an epithelial tissue thing.
Oh, okay.
Yeah.
I have a question.
Yeah.
I know you're going over the lecture notes. Are we eventually going to be able to ask you questions about the lab? Just because I know some of us are puzzled with what we have to draw and stuff.
Yeah, absolutely. So I'm going to go through examples as we get to the different types of epithelial tissues.
Okay.
I'll look at the examples from Dr. Lee's and from Wiley Plus, assuming that they have something for each and every different type. Dr. Lee doesn't have every single one, but I like how much you can zoom in on Dr. Lee's.
I actually like the WP more.
Yeah.
Yeah.
Like you can't zoom in as much, and I feel like the quality of the images isn't quite as good, but it's a little bit easier to navigate and see what it is exactly that we're talking about. And that's because it's geared specifically towards this textbook. I like to incorporate, I'm still happy to incorporate the Dr. Lee stuff just because it has just more examples and probably more real world examples. Like it's not always going to be like so pretty and easy to identify, you know, so it gives us a little bit more of a well-rounded collection of examples.
Okay, so you prefer us to do that instead? Like
no, not necessarily. Okay. When we get to the different examples, we'll look at examples, or when we get to the different types of epithelial tissue, we'll look at examples. And based on the examples and based on certain like identifying characteristics of the tissue types, you're going to basically be drawing like a, you know, whatever your artistic version or like a cartoon version of that particular like type of epithelial tissue, right?
Okay.
Like based on their characteristics, right? You can draw from a specific example, a specific image, but it's really more important to me that you can identify the tissue based on its characteristics rather than being able to identify which picture it was, right?
Okay. Perfect.
Yeah?
Yeah.
Any other questions?
No.
Good, okay. All right. Oh, yep.
So it doesn't matter like where we get our pictures, just that we understand it, like how you have it on your PowerPoints and you have it in the textbook, and you know.
Yeah, definitely. I'm not going to pull some random picture for you guys, like for your lab exam or something. Your lab exam will probably be pictures. It'll be identifying pictures, but it'll be stuff that you've seen before. So it'll either be out of this PowerPoint, It'll be out of your textbook, because now I have access to the e-text, so I can pull those images directly out. It'll be Dr. Lee's. It'll be a picture from your textbook, which will probably be a cartoon, a drawing, and the Wiley Plus histology stuff. But focus on the examples that we're going to be looking at today.
Okay. Yeah, that's because we were just trying to figure out like where are we supposed to like.
It's so much. I know, it's so much. This was a hell of a week. It was a hell of a week to miss, really. But hopefully we can get through it all.
Professor, I have a quick question.
Sure.
Okay, so just want to confirm the epithelial tissue, it doesn't have any free-flowing cells per se. It's just everything's tightly packed and the cells are just, they're immobile then, correct?
Yeah, exactly. Check out this picture in your text. This is actually, I was like trying to get over here so that you guys can see. Check out this example of epithelial tissue next to this example of connective tissue. In the connective tissue, these are the cells, and they're floating around in an extracellular matrix.
Wait, I can't see
Epithelial
it.
tissue, you can't see it?
I can't see it.
Oh, crap. What can you see in your screen?
You and Kim.
Oh, really?
Oh, she probably has our faces too big. You could minimize it. If you click on our faces, there should be like a list to minimize how we look.
I only see 5 of you guys.
Yeah, I only see 5 of us too.
Yeah, so in the top of that box where all of our faces are, change it.
Okay.
You can
I got it.
You got it?
Yeah, I got it. Thank you.
Okay, cool. All right, so you can see the PowerPoint now, right?
Yeah.
And if I switch over, you can see that I'm over at the textbook now?
Yes.
Okay, cool. So I'm back. So I'm over here in Wiley Plus checking out the e-text. There's this really cool example, figure 4.3. It's in your physical textbook as well, of connective tissue where you can see the cells are just free floating in an extracellular matrix, compared to epithelial tissue where all of the cells are packed together. They are all like side by side and touching each other. So this is epithelial tissue, super closely packed together, no really extracellular matrix to speak of, no extracellular fluid to speak of, but connective tissue is just cells like floating around in an extracellular matrix.
So I have a question though. Okay. Is that what makes it look kind of muscular? Is it just because they're tightly packed? It looks kind of like a muscle.
Epithelial tissue and muscular
like a skin
tissue, they can look similar. Epithelial tissue is going to have much smaller or like shorter cells. You'll be able to tell the difference between an epithelial tissue and a muscular tissue, basically because you're gonna be so good at identifying muscular tissue. So muscular tissue is looking, it's very distinctive once you've seen a few examples of it. Like skeletal muscle, it's very, very long. The cells are very, very long. There's lines. It's striated through those long cells. Even smooth muscle tissue has very, very long cells. So in epithelial tissue, the cells are closely packed together, yes, and on top of each other in some instances. But they are all like relatively short.
And then I want to confirm, so the microvilli and the cilia, they're basically the same thing. They're just
so they kind of look like the same thing from the outside, but they're technically not. So microvilli is technically like protuberances of the cytoplasm. Does that make sense? It's like bumps. It's like bumps on the top of the cell, on the apical surface of the cell.
Okay. It's thicker than the cilia, right?
Exactly, thicker than the cilia, versus cilia, which is like cytoskeletal elements. So it really is more like hairs coming out of the apical surface of the cell. And they are, since they're cytoskeleton and they have motor proteins, they can sweep.
Got it. So
there's nothing that moves them. They're just basically like bumps on the surface, on the apical surface of the cell.
But right? The only difference is their location, where they're located in the tissue.
Yeah, so that is a big difference. So you are going to have microvilli in places where you're going to be absorbing stuff. So you're like your digestive tract, your gut is going to have a lot of the microvilli on the apical surface of the cells lining that body, that organ, that part of your body. And cilia is going to be found in places where you're going to be like sweeping a substance across the surface of the cell. So either your trachea, where it's sweeping mucus up away from your lungs, or the fallopian tube, where it's sweeping the egg from the ovary to the uterus. That makes sense? Microvilli don't sweep, they don't even move. They're just there to create additional surface area at the top of the cell, at the apical surface of the cell, so that whatever that tissue is being exposed to, which is food and fluids, is going to get maximum, it's going to be able to absorb that at a maximum. Does that make sense?
Yeah. I got brush border epithelial cells of the small intestines where they facilitate the absorption.
Yeah, yes, exactly. Yeah, microvilli facilitate absorption, and cilia
brushes it off.
sweep. Cilia sweep.
Got it.
Yeah?
Okay, I had a quick question about Lab 9.
Okay, we're gonna get there. Is it general? Is it a general question?
Yeah.
Oh, shoot for it. Go for it. Let's see. I'll let you know if we're gonna talk about it.
So it's just about the drawings because it says
Okay.
to draw at high and low magnification, but since
It's gonna be tough, and we're gonna have to be a little bit flexible and resourceful about a lot of, I think, the instructions for the labs. So in terms of like high magnification, low magnification, I mean, you can do it with like the Wiley Plus examples. Like you can do it. It's not a big deal, but I would focus more on just being able to identify the tissue type. So like whatever magnification makes the characteristics for that tissue type clear to you is probably going to be the one that you're going to just go with.
Okay, so it wouldn't matter if we just drew like one picture that we understood.
That would be perfectly fine with me. Yeah.
Okay, thank you.
Yeah, no problem.
Question?
Yep.
Is this what we're doing for the rest of the semester?
Well, April 30th officially, and I wouldn't be surprised, honestly. I wouldn't be surprised.
Just because I did see the April 30th, but then it's like we're going back for 3 weeks. I don't know, it's just...
I know, what's the point, right? No, it would be easier if we just carried it through. I suspect that we probably will, anyway.
Ashley, I had a question.
Yeah.
So when it comes to the exam, it's still going to be the same schedule, right?
We're on the same schedule. What I'm going to do, so I don't want you guys to stress about exams. And I know it's totally freaky, and you guys aren't even like on the same schedule anymore. And we had a really good department meeting, Zoom meeting, last Friday, where we talked about what everybody's doing for exams. What we settled on was they're going to be online, obviously, but the idea is to have them open for an extended period of time, like 3 or 4 days. And they'll be unlocked for 3 or 4 days, and then you'll still only get your like 75 minutes or whatever, an hour or an hour and a half, depending, to finish the exam once you open it, right? But you'll have like 3 or 4 days to start the exam. That way, it's not going to be like, you have to have your exam at 2 p.m. on this day when you've got kids at home or you're working or whatever. So you can take the exam at any time in that window. Does that sound less scary? That be okay?
No, it kind of makes sense. Yeah.
Okay. I hope that will help.
Are you still gonna provide us with a study guide for the exams, or
yeah,
no?
Absolutely. Yeah, nope. I'm gonna try and do everything that I've been doing the same, like in terms of the content. Besides, obviously, we have to do this Wiley Plus instead of the hands-on labs. But everything else, like your syllabus, I'm trying to make it like it's still totally relevant, like in terms of like general scheduling and what we're going to be covering in the different weeks. This week is crazy. Obviously, we're going to try and do tissues and integument. But
someone asked in the chat, will the exams be held through Canvas or is there an option on Wiley Plus? Sorry.
Oh, we're going to do Canvas. Canvas is going to be our home base. The Wiley Plus is going to be strictly for like lab materials and extra study stuff for you guys.
Okay.
And that's it. I'm trying to keep everything like as the same as possible and on Canvas as much as possible. So the exams will basically be like extended quizzes, extended Canvas quizzes.
So is that what they mean by it being timed? Our exams being timed? We're basically
yeah,
just...
I think, yeah, probably. So you'll have, it'll unlock at one point, it'll lock maybe 3 days later, but at any point in that time period, you can start the exam and then you'll have an hour and a half to finish the exam. Does that make sense?
Yes,
Yeah.
thank you.
Is it gonna be like almost like when we're taking our quizzes, or is it? I know you mentioned it before that you didn't know if it was gonna be like recording us to see like what we're doing. Or
No, I still haven't decided whether I'm gonna use Lockdown browser, which basically keeps you from like
exiting the page.
Yeah, going to a new tab and like copying and pasting.
That works.
You know, like I know that you're still gonna have your textbook and your phone in front of you. It's just the way that it is. Like if you guys, it's kind of the honor system. Like if you guys are honest with yourselves about what you want to get out of this course and like actually learning as though this information is important, you know, which it is.
Let us all get A's, Ashley.
Yeah, I know, right? I know. But we are all in the same boat. Like all the faculty, you know, we're all in the same boat. If they're going to be using anything, then they're going to be using Lockdown browser, and that's really the only reason why I would do that is to keep anybody from copying and pasting.
Yeah.
That's all. That's really all that I can do, you know. So, yeah. So I will tell you a lot more about exams when we get a little bit closer. Any
Is
other
our
questions?
second exam still gonna be on the same day that's on the syllabus? I think it's like coming up soon.
At least it will start on the same day. Yeah.
Okay, just making sure.
Yeah.
I think we're all freaking out.
I know, we're all freaking out. Don't worry. Everybody is freaking out. We are all in this boat together. It's crazy.
I think it's because obviously it's like a science class and it's so important for all of us, especially if we're trying to get an A or like.
It's intense. I know. The balance between wanting to get the A to be competitive for applying to nursing schools versus like trying to actually absorb the information. Like it's a delicate balance. I'm trying to I think with Wiley Plus it'll be a big help. I think we'll still have like a really competitive course. And you guys have to take, there's like standard exams, right, that you take.
Yeah, actually they're pretty intense. I do like them. I mean, it does it's pretty cool. I kind of like it.
Yeah. So just because the exams will technically be open book, still like try and get as much as you can out of it for yourself. Just so that you have it for when you end up having to take those standardized tests, because you're gonna need all this information.
Yeah. But honestly, Ashley, you're doing such a good job, because a lot of my other professors are just like, let you know later. You know?
No, I feel bad. I want you guys to not be freaked out. Like it's like everything is already scary enough, you know, without.
I think you're making this really easy for all of us. I feel like, you know, you're implementing so many things and adding so many like websites. I feel like it is helpful because of
really, and not just overwhelming. I also didn't want to just like throw a bunch of stuff at you, you know. Be like,
we appreciate you
every day. Like, oh, and this. Oh, and by the way, this. Oh, and by the way, this.
I agree. We appreciate you.
Okay,
yeah.
Cool. Yay. Okay. I'm glad. I am definitely encouraged And I will like carry on this journey in the way that I have been until you guys tell me that I'm messing up. But any other questions before I keep going? I'm gonna keep going. Okay. All right. So still talking about epithelial tissue. We haven't gotten to the different types yet. We're just talking about these characteristics that are true of all epithelial tissues. They regenerate quickly, avascular but innervated. They're supported by a connective tissue basal surface or basal membrane. And they are all composed of tightly packed cells with some sort of like tight junction or gap junction or desmosome cell junction holding them all together, okay? So not really, no extracellular fluid, or I guess there is extracellular fluid technically, but not an extracellular matrix. If you're going to be identifying an epithelium, you are going to be asking 2 questions. So this is where I was talking about, so you can characterize them by location and function, but this is like how you're going to visually identify the type of epithelial tissue, is by asking first, how many layers does it have? Okay, so if it has one single layer, it is going to be a simple epithelium. And if it has multiple layers, more than one layer, it is a stratified epithelium, like the strata of like the rocks at the Grand Canyon, right? Like soil strata or something like that. Stratified epithelium has multiple layers of cells versus simple epithelium, which has one simple layer of cells. And they all touch that basal membrane, that basement membrane or basal surface. Okay, so that's the first question. How many layers am I looking at to find out if it's simple or stratified? The second question, I'm so sorry. Here's a picture. I do this every time. It doesn't matter that it's online now. I'm still doing this shit. Okay, getting ahead of myself. If for simple epithelia, you literally have one layer of cells, right? So here's the basal membrane or the basal surface. Out here is the apical surface that's open to the air or to the lumen of the hollow organ that it's lining. Simple type of epithelium versus stratified epithelium where you have multiple layers of cells, the bottom ones of which touch the basal membrane and the top ones of which make up the apical surface. And then you've got a bunch in between, right? And so this epithelial tissue, they're all touching each other, right? Okay. The second question, okay? First question, how many layers? Second question, what type of apical cell does it have? So what is the type, what does the cells look like that are like at that apical surface, okay? Are they squamous? Are they cuboidal? Or are they columnar, okay? So if we're talking about a simple epithelium, it can be simple squamous, it can be simple cuboidal, or it can be simple columnar. And that just means that it is a single row of squamous cells, which are just little flat pancake cells, a simple row of cuboidal cells, which are cube-shaped, or a simple row of columnar cells, which are shaped like a column, which look like a column. So they're like a rectangle standing on its end. If we're talking about stratified or multi-layered epithelial tissue, right, as per that first question, then you name it according to the apical layer of cells. So I'm going to go back a slide, right? So if we're talking about stratified, we're going to call it according to what the cells at the apical surface look like. So what these cells look like. Okay? Okay. So here's what a squamous cell looks like in cartoon form. It's like a little flat pancake cell. Okay? So simple squamous is a single layer of these flat pancake cells, and stratified squamous epithelium is going to be multiple layers of flat pancake cells. Simple cuboidal epithelium is going to have a single layer of cube-shaped cells. And stratified cuboidal is going to have multiple layers of cube-shaped cells, or at least the apical layer of the epithelium you're looking at is cube-shaped cells. And simple columnar epithelium is a single row or layer of column-shaped cells like these, and stratified columnar epithelium is, of course, layers of columnar cells, or at the very least, the apical surface of the tissue has columnar-shaped cells. Okay? Squamous, cuboidal, columnar. Simple or stratified? So those two questions. How many layers? Is it simple or stratified? And what is the shape? Squamous, cuboidal, or columnar is going to tell you whether or not you're looking at simple squamous or stratified columnar or simple cuboidal or stratified cuboidal, etc., etc. So let's talk about each one of those individually. And while we do, you'll want to note specifically where those cells are at in the body, what types of body parts you find those cells at, this type of tissue at, the function of this particular type of tissue, which is usually like, does it filter things, or is it for absorption, or is it just for protection, etc. The general description is going to hopefully kind of come with the name once you understand that stratified squamous epithelium is multiple layers of squamous cells, little pancake-shaped cells. You'll be able to describe it as such. Okay, so here is our first example. This is our first tissue type specifically. It is simple squamous epithelium. Okay, so you got a simple as in a single layer, squamous as in they are pancake-shaped epithelium. The function of simple squamous epithelium is to let things basically like diffuse and filtrate back and forth across this layer of cells. So because it is simple and squamous, it is a very, very thin type of tissue. It's going to make a very thin membrane, right? You've got diffusion functions, filtration functions. These are areas where protection is not important, since this is, of course, very, very thin and can easily be broken. It may secrete lubricating substances when in the serosae. So if you don't remember, the serosae are the membranes that line our body cavities. And they are made of simple squamous epithelium because it's a very, very thin membrane. So considering the diffusion and filtration functions of simple squamous epithelium, consider where it is found. The kidney glomeruli. This is a really super fun word for a little structure in your kidney that is basically, it's like it's a little ball where capillaries collect on the inside, and you have that simple squamous epithelium that is encasing the inside of that ball. And what's diffusing is the substances that you're pulling out of your blood to send out of your body via urine, right? In your kidneys. This is filtrating your blood. So this is a filtration function. The air sacs of your lungs. This is a diffusion function. So here's the picture that is in your, does your textbook have this one? No. So it's in your PowerPoint. And we can also look at an example of lung tissue if we were to go and look at Dr. Lee's histology. Okay, so here's Dr. Lee's histology. I'm gonna go to collections, general. We're gonna look at epithelial tissue. And here's some lung. Let's see, what else? Where else is it found, right? So we've got kidney glomeruli, kidney tissue, air sacs of the lungs, the lining of the heart, the lining of blood vessels. So again, diffusion, right? Because you've got nutrients and oxygen, carbon dioxide passing through those structures. Same with lymphatic vessels. You've got cells in that case. Other cells need to pass through easily, and it's really hard to do if it's not just one layer. And then the serosae, right? So the linings of your body cavity, of your body cavities. So this is lung tissue. We're looking at simple squamous epithelium. I'm just gonna skip over here to back to Dr. Lee. We've got some lung tissue here that we could look at. We've got, do we have any kidney? Move my stuff around. Oh, hello, there it is. Let's take a look at this, see if we can see a glomerulus over here. Okay, so I'm gonna show annotations. Sorry, I have to keep moving you guys around. Okay, so we've got some things highlighted. This is another reason why I really like Dr. Lee's. This is really cool. So let's see, what's that? Tell me, what is this? All right, so this is exactly what I was looking for. This is a glomerulus, and the lining it is simple squamous epithelium. So I'm gonna zoom in even more because I can, because this is awesome. Can I actually make the line go away? Actually, we'll look at the one right next to it. So this too is a glomerulus. Lining it, you can see a line of nuclei, okay? A single layer of nuclei. So we're looking at this as a cross section, right? This is like a slice of kidney. We're looking at a slice of a glomerulus, which is a little round sac, basically, of capillaries. And the lining of that sac, we're looking at it like down lengthwise, right? And we can see, what we can see is the nuclei of the individual simple squamous epithelial cells forming a single flat layer in the lining of this glomerulus, okay? If I take us back, ooh, rude, pardon me, and we look at lung tissue, and show annotations again. Oh, there's just this is that one little bitty one. Okay, so we're talking about the like the alveoli, or the little air sacs in your lungs. Again, diffusion of oxygen and carbon dioxide, right, needs to go directly through a thin layer of cells to the bloodstream. So we're talking about diffusion. And simple squamous epithelium is the name of the game there. So in this image, in this little example that they picked out for you, you can kind of see how it is. Well, one, it's very, very thin. You've got up here, it's same thing. You've got like a single row of nuclei, because we're looking again at a section, some slice of lung tissue. So we're looking at the simple squamous epithelium on a sideways, or like in a cross section. Does that make sense? So when you're looking for simple squamous epithelium, you're going to be looking for examples, or I will give you an example where it is clearly a single row of very thin cells, okay? The reason why you'll be able to tell that it's a single row of thin cells usually is by seeing the nuclei lined up, okay? So you see one row of nuclei lined up, then you know that it is a single layer of single layer, squamous, flat, pancaked for diffusion, right? It is epithelium, right? Because the cells are all butting up against each other, right next to each other, without like an extracellular matrix that they're floating in. Okay, simple squamous epithelium, epithelial tissue type 1. Are we okay with that? We okay with the examples? We can find an example from this one as well.
Yes, you've cleared up so many questions for me already.
Good, 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 good.
Professor, I'm sorry. I had
Yeah,
to
no.
leave for a quick second. Really
Okay.
quick. What is that white space?
The big white
Were you
space
able?
in the lung tissue.
Yeah.
This is okay. So your lung tissue, remember, it's your trachea goes down to bronchi to bronchioles. So it basically branches out like an upside down tree or like a root system. And at the very, very tips of those branches for each of your two lungs are kind of like, they're like bunches of grapes, except they're little hollow sacs, and they're lined by this simple squamous epithelium. The air that you inhale into your lungs fills up these little sacs. So each one of these big white spaces is an air sac lined by simple squamous epithelium, and it is butting up against connective tissue that is vascularized. So the blood vessels that the oxygen from the air that you breathe goes through that simple squamous epithelial tissue, because it's for diffusion, it's very thin, right? And goes directly into your blood. Does that make sense? So these are all like cross-sectioned alveoli.
Yeah, no, that makes perfect
Air
sense.
sacs
So
in
that,
your lungs.
got it. Because
Yeah.
epithelium is just the lining. That's
It's
basically
just the lining.
Got it.
Yep.
Thank you.
Exactly. No problem. So if I go to the module for chapter 4, tissue types, is that what I went into? Structure of the skin. That's not what I went into or wanted to go into. If I go into chapter 4, any one of these bits in the chapter 4 module, I can find, if I go to the Explore tab, Real Anatomy, which is the real reason why I wanted Wiley Plus. This is the cool stuff here. So here's the Wiley Plus Real Anatomy version of our histology lab. And we should have all kinds of good examples of this too. Artery and vein, ovary, looking for a lung or kidney. Mammary, hair follicle, seminiferous tubule. Are you guys feeling okay about simple squamous? Because I'll just move on if we're okay with it.
Yeah.
Okay.
Yes.
I think we're good. I might I think this might be for a different type of tissue. So instead of confusing you guys, I'm going to.
Are you looking for an example in the W?
Yeah, just so that you can see that there's
Oh,
one for Dr. Lee's as well as one in Wiley Plus.
So you go on, go back on the WP where it says the chapters.
Yeah, okay. Wait, let me see if I can do this. Oh, that's exactly what I want to do.
You go up, go to the chapters. You go down. And then right there where it says, yeah, right there, that one. There it shows you different kinds.
Specifically?
Yeah, up right there. This is two interactive.
Oh, I see what you're saying.
And then,
oh, excellent. That's cool. Yeah, so we've got let's see these. Okay, so we've got epithelial tissues. These is going to be more columnar or cuboidal. This one looks like, depending on what angle we're looking at that at, pseudostratified, simple columnar, simple squamous. It is.
Mhm.
Yeah. So that would be a good example of simple squamous, as long as you knew that it was like a top-down view.
You could click on it, it shows you more.
Excellent. Excellent. Oh yeah. Okay. So here's our glomerulus, or Bowman's capsule. It's another word for glomerulus. And our simple squamous epithelium is going to be lining it, so it's highlighted there. Oh, and it did a thing.
Yeah, go next to the simple squamous where it says on the side.
Oh, there it is. Yep. Cool. All right. So you can see how it is from the side in terms of a cross section, like this picture is, versus looking down at it from the top, where you can see that they are a flat layer of squamous epithelial cells. Cool, awesome, thank you. Is that you, Kimberly?
No, DJ.
Oh, thank you, DJ. Okay,
Yeah.
so simple squamous epithelium, right? I already kind of went over this. We're going to talk a little bit about endo versus mesophylium in a little bit. I'm not going to talk about it yet. I want to move on to cuboidal, simple cuboidal. Okay, so simple as in single layer, cuboidal as in cube shaped. So this is still a type of epithelium. Where this guy is found is places where secretion and absorption are super important. So we've got, in this picture, we've got, again, the kidney, but we're not looking at the glomerulus, we're looking at tubules now. So this is a place where we're going to be secreting stuff from the bloodstream back into the urine to be disposed of, and absorbing things out of the urine back into the bloodstream that we like changed our minds about. The kidney is really cool that way. He's got a lot of these tubules. This is like looking down the tubule, right? Like you cut it like this, a long tube, you cut it like this, and you're looking down it, and you can see that the walls of that tubule is made up of this simple cuboidal epithelium. So it's not as thin as the squamous, right? You can see that they are more cube-shaped or round even, but you can still also see that you've got a single layer of nuclei, right? You don't have like a nucleus here, a nucleus here. There isn't another one squeezed in there. You have a single layer of nuclei forming this layer of tissue, which is how you know that it is going to be simple cuboidal epithelium. So kidney tubules also in glands. So you have lots of glands, of course, that are going to be secreting stuff either like onto your skin or into the lumen of an organ or directly into the bloodstream, and more on that soon. Do we have, oh, so let's see if we can find in Dr. Lee. That's deferens maybe, since that's a tubule. Do we get simple cuboidal there? Looks more column-like to me. So let's see. Okay, well, we'll just go back to our good old kidney, see if we can find a tubule cross-section. There we go. All right, so here we go. Simple cuboidal epithelium labeled here. So in this big old cross-section of a chunk of kidney, if you like just take a slice of kidney, you've got the glomeruli, which are of course those Bowman's capsules, where all the capillaries are congregated so that that simple squamous epithelium that's lining it can allow water or toxins or whatever to diffuse out of the blood and into the structure of the kidney. That's going to have simple squamous epithelium. But later on, after the Bowman's capsule, it goes into a tube, and that tube is going to be made up of simple cuboidal epithelium. Hey, calm down, calm down. Gotta talk to it sometimes, you know? All right, so those tubes are lined by a simple cuboidal epithelium. Are you guys okay telling the difference between the simple squamous and the simple cuboidal just by looking at it?
Yeah.
Yeah, does that make sense? It's still one layer of nuclei, but you can tell that the cells they're fatter. They're not like thin pancakes, right? So this is more for like absorption and secretion kind of stuff. You need more of that surface area or more of that volume on the inside of the cell to do those kinds of actions. Secreting stuff out, like exocytosis, right?
Mm-hmm.
Or absorbing stuff in, endocytosis kind of thing.
So even though that doesn't look like squares, we could just tell by the way
it's cuboidal, right? Because they are like, they're chunky and roughly like the same size all around, so
Okay.
it's a little bit roundish. The simple squamous is clearly, the squamous is clearly very flattened, and columnar is going to be very clearly long and skinny, tall kind of stuff. The cuboidal is going to be in between. Going to be
Okay.
fat, chunky little guys without being very flat or thin, and also not being very tall and skinny like a column. Okay?
Yes.
Okay, cool. All right.
Will we have to know exactly where it's located? Like, where are they located?
So I would go ahead and take special note of where the locations that are detailed on these slides.
Okay.
For these basic slides, yeah. So for simple squamous, diffusion filtration, just know that it's found kidney glomeruli, air sacs of the lungs. Those are the most important. Most of these don't have very many more than like 2 or 3.
Okay.
So it shouldn't be that much, but yeah, you kind of gotta know where these different types of tissues are found. Yeah. So that's simple squamous versus simple cuboidal and where they are found. Any other questions? I'm gonna move on to simple columnar. Let's see if we can find an example of simple cuboidal over here, actually, before I go crazy over here. Oh, where was I just at? Oh, no, never mind, that wasn't. Oh, here we go. How about a gland? This is also found in glands. Okay.
You have to know the apocrine gland?
We'll talk about that. We'll talk about them more when we talk about the integument, so we'll get to them. As for right now, I'd say simple cuboidal epithelium, glands. Keep it general. Glands.
Okay.
So
Okay, there's another question in
yeah.
chat, Ashley. It says, Ashley, I have my kids in the background, but could you confirm if we need the lab atlas? Would you recommend it more now that we are online?
The atlas will be great for studying, especially if you're not used to looking at a computer screen to study, which some people are not. I would recommend the atlas for you. That said, it is pricey, like steep. It's like $120 for the lab atlas. So if you can't do that right now, the course as it is still I feel like we still have enough resources for you to be just fine without it. Not going to be required. It's going to be really, really cool for you to be able to see the tissue examples, some more examples of like tissue cross sections for this chapter in particular. The dissection of the rabbit, which we may not end up doing. It'll still be useful for identifying musculature, but we'll probably, if we don't do the rabbit dissection, then we'll probably do the virtual dissection here on Wiley Plus to learn the muscles. So it's just it'll just be it'll be like a luxury, be a nice luxury thing to study with in addition to our online resources, but not required. Is that, I know I'm still staying kind of vague, but it's really up to you, unfortunately. Sorry. It's not required by me, so it's up to you. Okay? Okay. So we're back in Wiley Plus. We're looking at the Wiley Plus Real Anatomy Histology. We're looking at a gland. Don't worry about what type of gland it is yet. We'll talk more about gland types in a bit. And we've got simple cuboidal epithelial cells. So we've got a single row of nuclei. These aren't like as cube-shaped as some of the other stuff that we have seen, but knowing that it's a gland. So if you know that this is a gland and you see a picture like this, you're gonna need to know that it's gonna be simple cuboidal. 2, again, when we get to columnar here in a second, the difference should be very, very clear between cuboidal and columnar. If it's not, then the pictures or whatever examples that I choose will be very, very clear. I'll make sure that the cuboidal is like relatively very cube shaped, as opposed to columnar, which are going to be very tall and skinny and possibly have cilia or microvilli associated with the apical surface of the tissue. Okay, so again, simple as in a single layer, of columnar as in column shaped, long rectangle shaped, epithelium, right? They're all closely packed together. So we've got round, some of the nuclei can be oval. So in the other epithelial types, generally the nuclei are round. And in simple cuboidal in particular, the nuclei are very round. In columnar, they may be oval shaped. So like in this cute little drawing, we've got long, sort of long, oblong nuclei going on in these cuboidals, or in these columnar cells. And then you may also be looking for other cell types to confirm that this is columnar. For example, goblet cells are a specific type of, it's a unicellular gland that is often found in certain simple columnar epithelium. And we will see examples of that in a minute. So, oh, that's not what I meant to do. So the function of simple columnar epithelium is absorption. So we're going to be looking for these types of cells, this type of tissue, in places where we are absorbing nutrients in particular, right? So this is going to be lining the digestive tract, right? All the way from your stomach to your anal canal, right? It also can be in other organs, non-hollow organs, like your gallbladder. Some glands have columnar epithelium, and the ciliated, the types of columnar epithelium that have cilia on the apical surface are going to be the ones that are lining your trachea and your bronchi, as well as the uterine tubes, right? So your fallopian tubes, where we're like sweeping the eggs towards the uterus, and then some regions of the uterus itself. So we've got two general types of this simple columnar epithelium. It can have cilia or microvilli on the apical surface, or it might not, okay? They come both ways. So this is kind of a depiction of what it would look like if it was non-ciliated. And this is an example of ciliated columnar epithelium. Sorry, the quality of this image just went to crapola. So we've got columnar epithelium here, whereas the apical surface is touched by all of the cells, and the basal surface is touched by all the cells. That's how you know that it's simple, right? It's a single layer. They are columnar. You have a single row of nuclei. That's also how you know it's simple. They are columnar, so the nuclei are, they can be oval shaped. So these kind of oblong nuclei is also an indication that this is columnar epithelium. And then of course the cilia is really going to be the dead giveaway in terms of this being columnar epithelium. There's no ciliated forms of squamous epithelium, and there are no ciliated forms of cuboidal epithelium, okay? This having cilia on the apical surface is a columnar epithelium thing. That's what they have sometimes. So, so let's look for some more examples of simple columnar epithelium. Starting with Dr. Lee's, let's take a look. The small intestine is probably a good place to start. Let's see what we got here. How about you? What are you? What's that? Simple columnar. Boom. I'm on fire. Okay. So you can see, again, a single row of nuclei. So we know that it is simple epithelium. These are very long, skinny, tall cells. And We've got cilia on the apical surface, right? Here's the basal surface connected to connective tissue down here. And here's the apical surface. This is like the lumen. This is the hollow inside of your, what was this? Small intestine, right? So this is columnar, simple columnar epithelium. You can tell it's columnar because long and skinny and has cilia in the apical surface. Okay, non-ciliated columnar epithelium. Let's see where that is. Where that is found. Do we even care? Is that even a thing in your book? Maybe not. Oh yeah, non-ciliated simple microvilli, gastrointestinal tract, stomach, many glands, and gallbladder. Ciliated respiratory tract unit. Okay. Okay. So let's look for ureter. Oh no, not ureter. I'm thinking uterine tubes, not ureter. Different thing. Small intestine, vagina? Probably not. Appendix? Let's look and see what Wiley has. Arterial epididymis. Here we go. Simple columnar epithelium. Oh. You gotta select the little hand if you want to move these around. Okay, so, yeah, I mean, these are okay. I'm okay with this. They're organized a little bit easier, but I just feel like it's not like as intuitive based on our stuff. So based on our lecture notes. Simple columnar epithelium forming the lining of the mucosa with the vascular lamina propria immediately deep to those cells. Okay, so the vascular area, I believe that they're talking about is going to be basically this. So you can see we've got what looks like blood vessels here. So this is vascular, this is connective tissue, right? The avascular epithelial tissue on top of it is simple columnar. You can tell because, again, you've got a single row of nuclei, right? And so because it's like the actual meat of the cells is extending up beyond the nuclei, you can tell that each one of these cells associated with each one of these nuclei is long and skinny. It's a little bit harder to see like the walls of the cell, the individual cell walls or plasma membranes, which is why I think it might be a little bit more confusing. This isn't so bad over here. You can kind of see the individual cells here. But you can still tell that it is simple columnar epithelium because of the single row of, again, oblong, oval-shaped nuclei in very long column-shaped cells, okay? So there's just a single layer, and each cell has an apical surface and also a basal surface. Okay? Simple columnar epithelium. Any questions? Okay. All right. Ciliated and non-ciliated. Okay. Pseudostratified. Okay, so pseudostratified is, of course, going to be, it's kind of a weird version of simple columnar in that each cell doesn't necessarily reach the apical layer or the basal layer. They're going to reach one or the other, though. So it's not technically stratified columnar because you really, it's like you don't actually have multiple layers of cells because, like, for instance, if you look at this guy right here, this cell is touching the basal membrane and not quite touching the apical membrane, and yet it's not exactly like a separate layer from this cell right here, even though its nucleus is down here, and this nucleus is up here, okay? So for these guys, we've also got some cilia. We've got secretion functions. They secrete mucus. Here's one of those unicellular glands that I was talking about earlier, the goblet cells, that is also an indication of seeing columnar epithelium. These are also found in simple columnar epithelium. Again, we've got ciliated and non-ciliated. Because it's columnar, it can either have cilia on its apical surface or it may not. The non-ciliated type can be found in the sperm-carrying ducts of males and the ducts of large glands. So this is like for secreting and then sweeping substances, secreting them out of the cells and then using the cilia to sweep them off to wherever that substance is supposed to go. The ciliated variety is the one that lines the trachea, so it sweeps the mucus up and away from your lungs, right? So your upper respiratory tract, so in your trachea. So let's see if we can find some examples. I knew that I saw epididymis, and I'm like super curious to see. There you are. Yes. So the epididymis is one of these sperm-carrying ducts that males have. Is that the little sperms? Oh my God. Look at all the little sperms. Sorry, I'm just like stoked about this. Okay, so this is an example of pseudo-stratified columnar epithelium. You can tell because there is not a nice single line of nuclei for you to find lining this duct. Instead, you see, you can tell that it's columnar still because there's no nuclei like up here, right? So you know that like the cell must extend all the way from this basal membrane to this apical surface, right, which faces the interior of the tubule. So we know that it's columnar because these cells are so long and skinny, but it's not simple columnar because the nuclei are not all lined up in a single nice row, right? It's pseudo-stratified because some of these cells, like this weirdo right here, is more associated with the apical surface of the tissue, right, than it is with the basal surface of the tissue. And really, most of these other cells are more associated with the basal surface of the tissue than they are with the apical, although they are. So here's some more examples of nuclei of certain ones of these pseudo-stratified columnar cells where the nucleus is down closer to the apical surface, right here too, this guy as well. But if it was cuboidal, then you would be seeing nuclei down here, right? It's obviously not squamous because these guys are not flat and skinny. They are definitely like long, right? Does that make sense? Are you guys okay with pseudostratified versus simple? It's mostly has to do with where these nuclei are located, right? And where the tissue type itself can be found, right? So again, this is the non-ciliated type. So these guys don't have cilia on their apical surface. The ciliated type is in the trachea. I don't know if we have trachea.
When you say duct,
Mm-hmm.
can you, it's like a tube, right?
A tube, yep, exactly, yeah. So glands have ducts, right? So going from the gland to secrete it to like the surface. So like your oil glands of your skin have ducts that empty out onto the surface of your skin. Things like your pancreas or organs that also like secrete like digestive fluids or biles or whatever into an organ, which is still technically secreting to like the outside of your body, right? Since we're a donut, they also have ducts, which are tubes that send whatever that substance is out past the apical surface. of that tissue.
Perfect.
Yeah? Okay. I want to know if there's trachea, mucosal lining of stomach, liver, rectal pili liver. Anybody see trachea or bronchi? Bronchus? Bronchus or trachea? Nope, 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 nope. This is not so useful because you're only seeing the actual lung. I don't think you can actually see. I don't think this is like actually, this is actually still just like the lung proper and not like the bronchus, which should be over here. Yep. Okay, so I guess it'll be, it's epididymis, but what do they call it? Sperm carrying duct. So pseudostratified columnar epithelium, the only example that I'm seeing for you guys is the sperm carrying duct, which was that example from the Wiley Plus. So between that and this picture here, which is actually a perfectly wonderful example of the ciliated type, right? So we saw non-ciliated in that epididymis, the sperm carrying duct. And here is our ciliated version, which is found in the trachea or upper respiratory. We also have a really good example of a goblet cell, which secretes mucus, which is pretty cool as well. We'll talk more about those when we talk about glands. We're finally gonna get to stratified, oops, pseudostratified columnar. Nope, we're gonna go to our stratified epithelia now, okay? Are we guys good on simple epithelium?
Yep.
Okay, squamous, cuboidal, columnar. Pseudostratified is like between simple and stratified. So we're going to now transition into the stratified versions, starting with back at squamous, okay? So we saw simple squamous, which would be a single layer of cells like this. Stratified squamous is when we have multiple layers, right? Where it's like we're looking at the strata of the earth, lots and lots of layers of, again, very thin pancake-like cells. So we're talking about the cell type on the apical surface, right? If you were looking down here at the cells that are on the basal surface of the tissue, what would you think that was? That would look more cuboidal to me, right? So remember, when we're looking at stratified tissues, you are identifying it based on the cells on the apical surface only, okay? So on the apical surface, we've got very flat pancake-like cells. So those are squamous. So we've got stratified squamous epithelium. So this is a very thick membrane, right? We've got a lot of layers here which offer good mechanical and chemical protection. So the function of simple or stratified squamous epithelium is often protecting underlying tissues, which are subject to abrasion, right? So your skin, namely, right? So you've got, so you know, in columnar we have ciliated and non-ciliated. For squamous, for stratified squamous, we have keratinized and non-keratinized. I know you guys must be loving this right now. So keratinized is basically like ones that have keratin, which in your skin, it's a very, it's like a waterproof, waxy, a hard substance that gives your skin and your nails and your hair their like physical strength and their waterproofness. Okay, was there a question? No? Okay, so the keratinized ones are going to be your skin, your nails, and your hair. Sorry, we're talking about epithelial tissue. Your skin only. Your nails and your hair are just pure keratin. They're not cells, they're just keratin, secreted by cells, by your skin cells. So the stratified squamous epithelium, keratinized version is your skin, basically. The non-keratinized forms of stratified squamous epithelium are found in the moist linings of your esophagus, which is your food tube, your mouth, and the vagina. So again, subject to abrasion, right? So it can be keratinized in the form of your skin or non-keratinized in moist internal linings that are subject to abrasion. Okay, so do I have, no. Let's look for some examples here. So starting with Lee's again, just cause we've got some skin here. Let's see if we can find some, who dat? Keratinized, stratified, squamous epithelium. Okay, so this is the apical surface of the skin in this picture. So this is the outside world right here. This is the stratified squamous epithelium. So you've got lots and lots of layers. Oof, sorry. Can I go any more? No more, that's it. So this is basically indicating the layer that is the stratified squamous epithelium. I would argue that it is from here to here, and over here we're talking more about the underlying basal membrane of that. Yeah. So from here to here, you can see the very, very thin layers. What this may be actually is the dead skin on the very surface. So here's some that's like sloughing off over here. But that's why we're not seeing any nuclei or very vague sort of light colored nuclei amongst this stratified squamous epithelium. And let's see if we've got, I'm sure we've got some over here. Some skin, perhaps. Mucus connective, mucosal lining. Pseudostratified columnar epithelium over here. What the heck was that? I missed that. So there's another good example of that. Oh, here's some skin. Anything else cool? Ooh, cool. Looky. Okay. Oops. God, I keep forgetting. Damn it. Does anybody know how to get out of this where it like it selects things and everything else is in black and white? Like, come back.
For these examples, are you going to pick one for like our exams? Or do we have to be like, you know, just because learn like skin or?
So, whatever I use for the exam will have to come from either this or Dr. Lee's, or your textbook. But whatever I choose is going to be, one, I'm going to tell you where, I'm either going to tell you what you're looking at in terms of like, this is a picture of skin. And then I'm going to ask you what type of cell you're looking at, and I'll make sure that it's very clear what it is you're supposed to be looking at. And, or I can say, here is some simple cuboidal epithelium. What body part could this be? And then that way you guys will have a good understanding from both angles where the type of tissue is located and how to identify it visually. Is that okay?
A question? Is
Yeah.
it going to be like a response or multiple choice?
So for the lecture exam, it'll still be multiple choice and true false. And for the lab exam, I'm gonna try and keep it as much like a practical as possible. So yeah, I'm gonna have a picture and it'll be free response. I'm going to put in as many possible misspellings of the word as I can. But since you guys are gonna have your book, misspelling shouldn't be as much of a problem.
I'm not using my book.
Oh, you're gonna go hardcore. Hardcore on it. If you it doesn't matter, because even if I put in a bunch of different like ways to misspell every word, it really doesn't make a difference, because I'll have to go through and look to see if there are other ways to misspell the word too anyway. So I'll have to go through the exams and see what you guys actually put for the answers. So it's still not as much work for me, because that way I only have to look at the wrong answers instead of having to grade the whole thing, which will be automatic for me, which is super nice. Silver linings for me. But yeah, for your response on the practical, as per normal for the class.
Are we still doing the group A and group B like times?
No.
All together.
Yeah, no, that died with the hybrid course.
Aw, rest in
I
peace.
know. It was fun while it lasted, wasn't it? So sad. I lost my skin. I had to go back because I was mad that I accidentally like selected a part of it. I didn't want to. More seminiferous tubule. I'm seeing more and more stratified squamous. Is that what we're talking about? Stratified squamous? What is this? Epidermis. Okay. This section is from the mucosal lining of the vagina. This is a really nice one for stratified squamous epithelium. So you can see that we've got multiple layers of flattened pancake cells. Judging by the apical surface of it, we can tell that they are squamous, right? The cells down here are starting to look more cuboidal and then columnar towards the basal surface. So that's why you're going to identify it by what's on the apical surface. Is everybody clear on stratified squamous epithelium and how to identify it?
I have a quick question. So
Yeah.
for the bottom by the basal, if I'm correct, that it's always going to be cubital or look cubital?
Not necessarily. When you are looking at a stratified, when you can tell, first of all, that it's not simple, right? It's not a single layer. You'll be able to tell that first. If you, then you can eliminate this. But if it's a stratified, if you're looking at multiple, you just know that you're looking at a multi-layer epithelial tissue. Make sure that you are looking, identify the apical surface and look there to see what type of cell it is. So it's going to be stratified, but based on the cells up here, it's going to be stratified squamous. The cells down by the basal membrane, I think it varies, but judging just from like what we've been looking at, the examples that I've been giving you, they do kind of tend to look a little bit more cuboidal going into columnar, going towards the basal membrane. I think that's just because these cells are busy dividing, honestly. So it looks that way. And they get flattened as they go towards the apical surface because the cells are dividing down here. Remember, because epithelial tissue regenerates fast and a lot. So there's a lot of cell division happening at the basal surface of this tissue. So because the blood supply is down here with the connective tissue, right? Because epithelial tissue is avascular, that blood supply is feeding the cells that are currently actively dividing. And as the cells actively divide on the basal layer, they push all of the other cells up towards the apical layer. These ones die and slough off, right? Which is why we have like, that's why bed bugs like dead skin cells and nasty things like that, right? Because we're constantly shedding skin cells everywhere. So those shed and new ones come up. That's how we heal, right? And that's how we maintain that important protective barrier of our skin and of our esophagus, and if you're female, your vagina, right? To try and like keep those surfaces protected, right? Any other questions?
Can you hear me? Hello?
Yep, what's up?
I have a question. So is the function of the columnar and the cuboidal the same?
Sometimes.
Because it was saying like both of them had like secretion of like absorption or
Yeah,
something.
secretion and absorption are both a big thing in both of those types. So let's look again. Secretion for pseudostratified, absorption and secretion for simple columnar, secretion absorption for simple cuboidal. So yeah, and so you'll find them in similar places in terms of, you'll find them in glands. So yep, excretory ducts of some glands for simple columnar, and we've got ducts of large glands for pseudostratified columnar. So yeah, some of these do overlap, and they overlap in function as well. Yeah, it's true. It's true.
So besides the shape
Mhm.
of the cells itself, like where the nucleus is and the layer of the tall cells, what is the main difference between the two? Like how can we study like to distinguish between the two? What he just asked, the
like
columnar.
between columnar and cuboidal?
Yeah, since they're found in the same locations.
Yeah, so basically if it's columnar, you'll know that it's either going to be ciliated or non-ciliated. If it's ciliated, then you know it's columnar. First of all, it can't be ciliated. If it's cuboidal, it can't be ciliated. If I were to tell you for a non-ciliated type of columnar, like what would be like this? What would be the craziest one? What would be the most evil one for me to do, right? It would be like a kidney tubule, a duct, and a duct, right? Or something like that. And just be like, this is a gland. What kind of cells are these? Like, that would be mean. I wouldn't do that to you. I would
have failed.
I know that I would have failed. I wouldn't be able to do it either. That'd be messed up. I would be specific about what organ we're looking at. So like if it's a lining of a digestive tract, that's not found in cuboidal. Cuboidal is going to be, it's kidney tubules and secretory ducts. So if we're looking at like the lining of any part of your digestive tract, can't be cuboidal, like just rule it out. Kidney tubules, that's really, let's call that the only place that cuboidal epithelium can be found. If I'm going to ask you about simple cuboidal epithelium, I'll show you a picture of a kidney tubule. I'll write that down so I have it.
And then I had a question about the columnar.
Yeah.
So some of them do have cilia and some don't.
Mm-hmm.
Okay. Okay. In the first photo you showed us they did, and the second one they didn't. Correct?
So in this picture, which was just a random and not a very good one, it does show cilia. The ciliated, what did I have? So non-ciliated, because they have microvilli, right? They don't have cilia in your digestive tract. Okay. I hear somebody who's pissed back there. So this is your digestive tract. This is going to be your small intestine. Tall and skinny, the nuclei line up nicely, microvilli. Okay, so what you're seeing on the surface here is microvilli. So it looks a lot like cilia. So let's see if we can find something else here. Okay. That's like clearly cilia. What did I pseudostratified mucosal lining. Oh, so this is also going to be microvilli, not cilia. Cilia again is for sweeping, right? So we should be seeing that in like the trachea or the fallopian tubes, uterine tube. I'm just reading up to see. I keep wanting to like scroll to zoom in, and I keep doing that because I want to just grab it and go. Okay, whatever. This, whoops, oh, how cute. So you can actually highlight the cilia specifically here. So this is, again, uterine tube, also known as fallopian tube, whereas the lumen is where the egg passes through, and all of these cilia on the inside of lining the inside of this hollow tube is going to be like sweeping in one direction to keep the egg moving in the direction of the uterus, right? So this would be a really nice example of ciliated columnar epithelium, and since it is ciliated, you know that it's columnar, no matter what, right? Whether it's simple stratified, I wouldn't use this as an example of simple stratified because then all these nuclei are confusing. Let's see.
Ashley?
Yeah.
The thing kind of Liz was on, but it like took her out of the chat and she can't get back in.
Shoot. Back into our discussion? Into Zoom?
Yeah.
Can't get back onto Zoom? Well, that's lame. Shoot.
So
well, if you're in touch with her, tell her that I'm gonna finish up epithelial tissues and that it will be posted. It'll be recorded and posted. And what I'm gonna do is we'll take a little break after epithelial before we go into connective, and I'll like see if we can I'll find a link for her or something and see if we can get her back on.
Okay.
I wonder how many other people are having this problem though, is what I'm wondering. I know that Confer-Zoom has, it's been okay, but it's been struggling. It's been struggling.
Thank you.
Yeah. Yeah, see, like this looks like stratified columnar to me, but. Like in your book, it's amazing. The lining of the pharynx looks very, very similar to this, which is supposed to be the lining of this is supposed to be the uterine tube, right? Yep. Well, if there's any question, I'm not going to use the image, okay? Pseudostratified, take a look at this picture here. The ones that are in, at least the Wiley ones, are they're pretty good. They're pretty clear. Wonder, can I look up? Nope, I can't. This is the trachea. Oh, cool. Okay, so yeah, that's tough. The question between being able to identify between the different types of columnar, is that the question?
Yeah, that or the simple columnar. But you kind of answered it with the one with the cilia.
Okay.
Another one has microvillia.
Yeah. Yeah.
So that
yeah.
kind of made sense.
Okay. It's like they either, so if it has cilia or microvilli, it's going to be columnar. Whether it's simple pseudostratified or stratified, really there is not a great way to differentiate based on where they are located. since they're often found in similar regions, so simple columnar, non-ciliated in the digestive tract because there's microvilli, and then ciliated in ducts and your trachea, uterine tubes. Your pseudostratified, oh yeah, sperm-carrying ducts. That was a really good one, actually. But then again, trachea. So I'm not gonna give you a picture of a trachea and ask you if it's simple or pseudostratified unless it's like super clear that there is one row of nuclei. So whether it's simple versus pseudostratified, look for that one layer of nuclei versus having nuclei kind of all over the place, not in 2 layers, right? Because there is such a thing as stratified columnar, which we're going to talk about in a little bit. And yeah, I'm gonna move on. Is that okay?
Yeah.
Any other questions? Okay. All right. So we talked about stratified squamous, multi-layers squamous cells. Stratified cuboidal is also a thing, of course. It tends to be rare, so there's only a couple of places where it's found, and that is in the walls of certain types of sweat glands and mammary glands. So if I tell you it's a mammary gland, it's going to be stratified cuboidal epithelium. It's really the only place where it's found, and you can tell what it is by the 2 layers of nuclei present versus the single layer of nuclei present in simple cuboidal epithelium. Right? We've got multiple layers of nuclei here in the stratified cuboidal epithelium. So again, this is not it's not columnar because we've got like a cell here clearly and a cell here clearly, like they're not like slipping past each other. It's definitely one like sort of chunky fat cell with a nucleus and another chunky fat cell with a nucleus here. And it's only found in mammary glands as far as you guys are concerned. Is that okay with you guys? Stratified cuboidal,
Yeah.
call it the mammary gland one and be done with it? Okay. All right, so this is not what I meant when I said that. I think this is supposed to be stratified columnar. Sorry, guys. Strier, you're stratified. Okay. My bad. Stratified columnar epithelium, not simple. Hence the multiple layers of nuclei visible here, okay? So we again have columnar epithelium. They are long and skinny. They, the ones on the apical surface are, of course, touching the apical surface, and the ones touching the basal surface are only touching the basal surface. They are also in your pharynx, so isn't that lovely? We have simple pseudostratified and stratified columnar epithelium, epithelia in our pharynx, which is like your trachea. The male urethra, which is your pee tube. And of course, again, like so many other things, in the ducts of some glands. So let's see, also occurs transition areas between the 2 types of epithelial. Between 2 other types of epithelia. Oh, don't worry about that. Let's see some examples. Let's see if we can find some examples of stratified columnar. Urethra, excretory ducts, esophageal, mixed membrane, conjunctiva of the eye. Let's see if we can find some. See if we can find a urethra. Did we have a urethra? Ureter? That might work. What's that? Transitional. Nope, that's what we're going to talk about next. Well, let's see what Wiley has. Not uterine, ureter. I know we just did that. We want the, is that what we want? No, we want urethra. Esophagus. Ooh, maybe. Stratified squamous. Nope. Sorry guys, I want to find you a good example before I go crazy. Esophageal glands, I feel like it should be there, but it's not. If I go back over here, can I find it? Well, that's lame. I wonder if there even is an example of stratified columnar epithelium. I'm sure that there is one in here somewhere. I'd have to dig around. I don't know. I don't know, you guys. It's lame. I'm gonna have to get back to you guys on that. I'm sorry. Stratified columnar. I don't see a good example, so I'll have to find one for you and I'll email it to you. Strat column example. Okay. All right, so I'm gonna find an example, a better example than this for you guys of stratified columnar epithelium. It is a little bit tricky to identify the difference between the stratified columnar versus the pseudo-stratified columnar. The stratified columnar, remember, since it's stratified, we are identifying it based on the cells that are touching the apical surface of the tissue. So because these guys are long and skinny, but we've got 2 rows of nuclei versus the kind of funky everywhere kind of nuclei of pseudostratified, that's really how you're going to identify it as being a stratified columnar. And I would not be able to make you identify it via these because there are other types of columnar, specifically epithelium, that are also found here. So I'd have to find another way or another example to test you guys on this. If I find one, I will let you know, and if not, then I will make it clear on the study guide what exactly I want you guys to know about this. Okay. So transitional epithelium is the last. Oh, no, we've got glandular too. Transitional epithelium, before we get to the glandular stuff, is epithelial tissue that looks like stratified squamous and stratified cuboidal put together, and at different times. So the whole reason that transitional epithelium exists is to be super duper stretchy. Okay, so it looks cuboidal when it's relaxed, and then when it stretches out, it looks like squamous. So it's transitional because it transitions between looking like cuboidal and looking like squamous, depending on whether or not it's relaxed or stretched out. This stretching ability makes it most useful in the urinary bladder and associated structures like the ureters. So that's why that example over here of the ureter, which is the tube that connects your kidneys to your bladder, that tube is called your ureter, and it is lined by by transitional epithelium, because it needs to be able to stretch as the fluid travels through it. Same thing with your urinary bladder, right? That thing needs to be able to hold a volume of liquid, so the lining of it needs to be able to stretch. So when your bladder is empty, the transitional epithelium lining it is going to look like it's cuboidal or stratified cuboidal or something, which you know it's not, right? Because stratified cuboidal is only found in mammary glands and some other like random sweat glands or something, not in your ureters or your bladder. So this crazy looking tissue, keep doing that, epithelial tissue lining this structure looks like it could be cuboidal based on the cells that are at the apical surface, right? Because they look like they're not tall and skinny like columnar. They're kind of flat and pancake shaped like squamous. And then some of them they look like they're cuboidal, right? So it looks like they're either cuboidal or squamous. You're going to be able to identify this most easily by where it is found. If it's in the urinary bladder, if the example is the urinary bladder, then it's going to be your transitional epithelium. Do I also have, no, what's this? Urinary mucosa. So more, so this is the urethra that we're seeing here. And again, at the surface, these look like, since we're identifying a stratified epithelium, these look like they could be cuboidal, right? So it must be relaxed. And when it is stretched, these guys will flatten out and look more squamous. So I won't be able to, I'm not gonna like try and trick you and be like, hey, like, what is this? Just based on looking at the cells themselves. Even though we know that stratified cuboidal doesn't exist anywhere except for mammary and certain sweat glands, this transitional epithelium is going to be identified based on its location. So lining of the bladder, the urethra, and the ureters, which are the tubes that go to and from the bladder, respectively. You guys okay on transitional epithelium?
Yes.
Okay.
And you said that we could, we're gonna post it so we could still go back and just, you know, you just said, right? We could rewatch. Okay.
Yes. No, I'm recording this and I'm going to upload it sometime tonight. So this is going to be the epithelial set portion of this lecture, and then I'm going to go through, I'll basically record separate or split it up between the different tissue types. This will be the epithelial one. It's going on a couple hours long here. So it'll be a quite large enough video on its own. So I'm going to stop the recording and restart it before we go into connective tissue. Okay, so let's talk about the last epithelial tissues, which are the glandular epithelia. I don't want you guys to know or be able, you don't necessarily have to be able to identify this by sight. Just know that you've got, this is really more of a rundown of gland types. So we've got two different generally types of glands, endocrine and exocrine glands. Endocrine glands are they don't have a duct, okay, so they don't have a tube associated with them. Instead, they secrete their product directly into the bloodstream. Examples of endocrine glands are like your thyroid gland and other like internal body glands that aren't including like your pancreas, which has a duct that goes into your digestive system, which is technically outside of your body, right? So endocrine glands, endo as in like inside, they don't excrete things through a tube to the outer surfaces of your body, whether that be your skin or your the inner lining of your digestive tract. So they can be unicellular or multicellular. The only kind of unicellular gland, it's an exocrine gland, and it's the only one we're going to talk about, that is goblet cells. So again, those ones secrete mucus. Let's see what we've got here. So endocrine glands, again, they are ductless glands. This is not a duct. This is hormone precursor. So this is basically like the product, the product of the gland that will be moved into the bloodstream via not a duct, via not a tube. So it goes directly into the bloodstream. There's probably like a blood vessel somewhere where it can like diffuse through the wall of the blood vessel through a simple squamous epithelium. And that hormone, which is being stored here in this bubble, will leave the endocrine gland that way instead of going out through a duct to an external surface of the body. So this is an inside, this is a cross section of a piece of somebody's thyroid gland. And it would be a thyroid follicle. And this is just the hormone that it produces. This is not looking down a tube. We're not looking down a duct. This is a bubble, right here, okay? So the hormones that it produces travel through lymph or blood to the target organs. So no duct. Exocrine glands, in contrast, looking at one from the side, right? The gland is here, and then you have ducts. You have a duct that travels to the apical surface of the epithelial tissue and is an exterior surface of your body, or a surface that is exposed to outside elements. So that includes your digestive tract or your skin. We have many more exocrine glands than we do endocrine glands, mostly because your skin is just peppered with like sweat and oil glands, right? And then all of the glands of your digestive system, which are secreting mucus or digestive enzymes and all kinds of other stuff. Those are all exocrine glands. So they secrete their products into ducts. This is looking down into a duct. Okay, so we're looking down into a tube here. We've got what looks to be simple columnar epithelium here lining these ducts of this gland. So the secretions that exocrine glands secrete go directly from the cells lining the gland itself and the ducts of the gland, and they excrete those products directly into the tube, into the hollow middle of the tube, so that they can be secreted out onto the surface of the skin or into the lumen of the stomach or the small intestine or the rectum or whatever. So mucus, sweat, oil, and salivary glands are all exocrine glands. They all have ducts, okay? Unicellular exocrine glands are ones like this, or I'm sorry, multicellular exocrine glands are ones like this, where you have many, many multiple cells that make up the gland and line the ducts. There are, however, unicellular exocrine glands. The one and only example that we're going to talk about is the goblet cell. So these in blue here are goblet cells. They are interspersed among this simple columnar epithelium, and they secrete mucus out onto the surface or into the lumen of whatever body organ this is on the apical surface of the this epithelial tissue, of this simple columnar epithelial tissue, okay? Here's another picture of a goblet cell. Isn't it beautiful? It's just one single cell that secretes mucus, and it's like, gets sandwiched in between other epithelial cells. It's just a type of exocrine gland. Kind of cool, pretty weird. Multicellular ones are the rest of them. So they have a duct and a secretory unit. So like this would be a secretory unit, and they all feed into the duct that leads to the surface, okay? We have several different like shapes of multicellular exocrine glands. They can be simple or they can be branched, okay? I'm not worried about the rest of these types. Just know that they can be simple, as in one little secretory section and one duct, versus branched, which is multiple secretory structures and at least one duct. I guess they all are going to lead to one single duct on the surface of whatever it is that they're secreting or excreting onto, right? So just focus on these simple duct structures. Of the branched, you can have compound, and then there's also slightly different types. And they're not even like, there's not even an important example of this one in humans, so whatever. Exocrine glands can be simple or they can be branched, okay? And there is a lot of variation to this branch, to these different branch types. Do I care? Do we care? Am I worried about this? Are we moving on yet? Okay, almost there, you guys. Hang in there. We've got different ways that secretions are produced or secreted by exocrine glands. They can be secreted by exocytosis. So that is, of course, within a cell, producing the substance, then releasing it into the outer environment by the joining of a vacuole on the inside of the cell with the plasma membrane, and then opening up that vacuole to the outside of the cell. Pancreas, sweat, and salivary glands are merocrine glands, which is what we call glands that secrete their products by exocytosis. Holocrine glands are exocrine glands by which the products are secreted by rupturing the gland cells themselves. So sebaceous glands, sebum is an oil, right? This is the stuff that gives you pimples, right? So it's those sebaceous glands in your skin that get clogged up. They produce that sebum, which is the name of the oil that sebaceous glands produce, by the gland itself rupturing and releasing its product into the duct of the exocrine gland. And apocrine glands are somewhere in between So basically you kind of have, some of them are secreting their product by exocytosis. Some of the cells of the apocrine gland are secreting by exocytosis, and some of them are rupturing on the surface, on the apical surface. Some of them are rupturing. So apocrine sweat glands, which were mentioned earlier, are so called because of this feature, because of their mode of secretion. So here's your merocrine gland with the green secretion being produced by the cells individually, releasing it by exocytosis. Here's a holocrine gland whereby the cells themselves are detaching and rupturing, and it's the whole disintegrated cell and its content that is the secretion. So holocrine glands, such as sebaceous glands, is that what he said? Yeah, such as sebaceous glands, they actually release the entire cell into the duct with all the content, and that all of that is the secretion. But it's why they get clogged up so much, right? That's why those sebaceous glands get clogged up so much. And then apocrine glands, they kind of do both, okay? So you've got some of this exocytosis action happening, but also on this apical level of the cells, parts of those cells also disintegrate and pinch off, and that forms the secretion. Okay, so any questions about glands? Merocrine, apocrine, holocrine. Endocrine and exocrine. Apocrine, holocrine, and merocrine are all types of exocrine glands, right? They all excrete something into a duct. Endocrine glands are like your thymus, your thyroid gland, a lot of your other like internal bodily glands that don't secrete a substance into the lumen of a hollow organ or out onto your skin. They secrete directly into the bloodstream, endocrine glands.
Actually, I have a question.
Yeah.
Is there already due dates for lab 6 through 9, or you still
6 through 9, I'm having due on Thursday. But if that's not enough time for you guys, I can extend that. And
That would be nice, just because we didn't know what the heck.
I know. Well, yeah, it's like it was kind of a crapshoot to see whether or not how you guys would do with this, like without any direction at all. It's like, well, they've got the chapter, like let's see like what happens. If you guys want more time, that's fine with me. I'm here to be flexible for you guys at this point.
We have pictures of like our notebook, our lab book to like submit the assignment.
Sorry?
Do we have to like take pictures of our lab book and then submit it?
Is that okay with you?
Yeah, that's fine.
Okay, yeah, that was what I was thinking. Yeah, just take pictures of it, or if you want to like scan it in with a scanner and send it as like a PDF or an image file or whatever, that's fine too. But I figured it would be easiest for you guys if you just want to like take a picture with your phone and send me that picture. Yeah, just upload or submit those into the assignment on Canvas. So
yeah, there's some questions like in the lab, like do we have to answer all of them? There's just like some that are like,
One asks, how many different samples do we have among the lab specimens?
You can't. Yeah, there are some of them that you're not going to be able to answer, because the ones that are like specific to the lab protocol that we would have been following if we were in person, you can't answer. Everything else that's like specifically like theory that you can answer, do. It's gonna be those ones. So.
Okay, yeah. That's what I was like wondering. I don't know.
Yeah, don't sweat it. Don't sweat it. I want to give you guys like the support and the structure that you need to learn what you need to learn. But there's going to be a lot of flexibility for things like that, and grading and stuff. So while it might make the class easier in some respects, I don't want you guys to like lose sight of like the main goal, which is to learn the material.
Yeah, of course.
So it sucks like a lot. I feel like to not be able to see you guys in person puts a lot more of that burden on you guys. And I want to give you guys as much structure as possible. It's just not as possible anymore as it once was.
Yeah.
Yeah.
And we were doing so good.
I know, we're getting there. We're getting there. We were having a good time, weren't we? But it's still gonna be fun. It'll be cool. So let me pause this recording. I guess I have to pause share. Ugh, fine. It's gonna go back to my face. I hate it. Okay. Whatever. I'm gonna stop the recording.